the utricle. This is the cochlea, the semicircular canal, the vestibule, the utricle, and the saccule. That is the utricle, and that's the saccule. The vestibule of the ear encompasses a portion of the inner ear roughly 4 millimeters in size. It is a bony cavity within the temporal bone that contains organs and nerves associated with the vestibular system. It is posterior to the cochlea, I repeat, posterior to the cochlea and anterior to the semicircular canals, the vestibule of the ear. That is the vestibule of the ear, posterior to the cochlea, anterior to the semicircular canals. The vestibule is an approximately 4 millimeter chamber dominated by depressions housing parts of the membranous labyrinth. Now these are the utricle, the saccule, that is the utricle, this is the saccule. The utricle is an elliptical recess, the saccule is a spherical recess. The basal turn or the end of the cochlear duct, which is the cochlear recess. It is located medial to the tympanic cavity, posterior to the cochlea, anterior to the semicircular canals, and in the lateral wall is the oval window with the stapes footplate covering it. The cribrous areas have perforations through which the nerve bundles gain access to the inner ear. The endolymphatic duct is housed within the bony vestibular aqueduct and originates at the postero-inferior aspect of the vestibule, that is via the utriculosaccular duct. Now the utricle is a small membranous sac and paired with the saccule lies within the vestibule of the inner ear. It has an important role in orientation and static balance, particularly in horizontal tilt. The vestibule is located within the bony labyrinth of the inner ear. The utricle and the saccule are co-located within the vestibule. The utricle lies on the medial wall of the vestibule and is in contact with the recessus ellipticus. It is larger in size compared to the saccule, and the utricle communicates with the saccule via the utriculosaccular duct. Lying horizontally on the floor of the utricle lies a projection known as the macula, the sense organ of the utricle. Now, this sensory epithelium is lined by vestibular cells, which are mechanoreceptors containing stereocilia and gynoecilia. Now, these cells project into the membranous labyrinth called the statocorneal membrane, a gelatinous layer beyond containing otoliths, which are calcium carbonate particles. Cilia from the hair cells are attached to these otoliths. Function: any change in the orientation of the head results in stimulation of the utricle via movement in the otolith cilia macula complex, releasing signals that the brain interprets. The utricle is more sensitive to horizontal acceleration in comparison to the saccule, which is more sensitive to vertical acceleration. The utricle is larger than the saccule. It is of oblong form, compressed transversely, and occupies the upper and back part of the vestibule, lying in contact with the recessus ellipticus and the part below it. The macula of the utricle is a small thickening 2 by 3 millimeters lying horizontally on the floor of the utricle, where the epithelium contains vestibular hair cells that allow a person to perceive changes in latitudinal acceleration as well as the effects of gravity. It receives the utricular filaments of the acoustic nerve. The hair cells are mechanoreceptors which have 40 to 70 stereocilia and only one true cilium called gynocelium. The gynocelium is the only sensory aspect of the hair cell and is what causes hair cell polarization. The tips of the stereocelium gynocelium are embedded in a gelatinous layer which together with the statocornea form the otolith membrane. The membrane is weighted with calcium carbonate protein granules called otoliths. The autolytic membrane adds weight to the tops of the hair cells and increase their inertia. The addition in weight and inertia is vital to the utricle's ability to detect linear acceleration and to determine the orientation of the head. When the head is tilted such that gravity pulls on the statocornea, the gelatinous layer is pulled in the same direction also, causing the sensory hairs to bend. Labyrinthine activity responsible for the nystagmus induced by off-vertical axis rotation arises in the otolith organs and couples to the oculomotor system through the velocity storage mechanism. An otolith, also called statocorneum, is a calcium carbonate structure in the saccule or utricle of the inner ear, specifically in the vestibular system of vertebrates. The utricle and saccule in turn together make the otolith organs, and these organs are what allows an organism, including humans, to perceive linear acceleration both horizontally and vertically, and they have been identified in both extinct and extant vertebrae. Now these are the otocornea, statocornea. Look at their configuration and how they are nicely chiseled and in its shape, hectagonal shapes, and how they are located within the utricle and the saccule. Basic assumption for functions of otocornea. From a medical point of view, it is generally accepted that otocornea couple mechanical forces to sensory hair cells in order to detect linear acceleration and gravity for the purpose of maintaining bodily balance. Human otocornea are formed at late embryonic stages and normally undergo only minor changes with lifetime, although some maintenance after formation may be required. Head tilts and linear head motion causing displacement of the otocornea array, namely tens of thousands of individual otocornea, producing a shearing force which deflects the underlying sensory hair cells in the sensory epithelium, namely the macula. Anchoring of otocornea is essential for optimal vestibular function and balance. Now look at these otocornea, and they are anchored by thin fibrils. Let us see what these fibrils are made of. Otocornea are not only anchored to the sensory epithelium but also interconnected by a network of protein fibrils. These are the networks of protein fibrils as shown. On the one hand, the protein fibrils represent the organic component of otocornea, which is integrated into the composite system. On the other hand, the fibrils extend over the volume of otocornea into the surrounding endolymph, thereby forming the interconnecting net. These otocornea are embedded within a fluid called the endolymph. The chemical composition of the fluid medium surrounding the otocornea has been investigated, and quantitative data, though not available, the qualitative presence of the following components is much certain. The endolymph primarily contains proteins, sodium, potassium, calcium, chloride, and hydrogen carbonate. The pH value condition is around the neutral point of 7. A significant viscosity of the endolymph is assumed to be caused by the presence of specific glycoproteins. What causes imbalance or inner ear toxicity? By gentamicin. Gentamicin causes irreversible structural damage of the otocornea. Today now we know why it is ototoxic, and it does so by progressive dissolution of calcite. The sequential scanning electron microscopy images presented impressively verifies the complete dissolution of calcite component of human otocornea within minutes. And here we see it is a body that gets diluted, and after 10 minutes of exposure, The chemical attack starts in the belly area and extends over the branches until the autoconium is completely dissolved. The images clearly reveal that autoconia as solid composite specimens are threatened by chemical attacks in general. Stereocelium. Stereocelium are non-motile apical cell modifications. They are distinct from cilia and microvilli but are closely related to microvilli. They form single finger-like projections that may be branched with normal cell membrane characteristic, and they contain actin. Stereocilia are found in the sensory cells of inner ear. Gynocelium. These are the stereocelium, but there is one long strand, and that is called the gynocelium. Gynocelium are specialized primary cell present in auditory hair cell in the inner ear. These cilia do not directly mediate auditory mechanoelectrical transduction, but partially retain the characteristics of motility responsible for the response of hair cells to sound stimuli. Mechanoreceptor for hearing and balance, the vertebrate hair cell, the modified epithelial cells, cilia on apical surface, we have the gynocelium and the stereocelium. One gynocelium is the longest cilium located on the hair cell next to 40 to 70 stereocelium. During movement of the body, the hair cell is depolarized when the stereocilia move towards the gynocelium. The depolarization of hair cell causes neurotransmitter to be released and increase in firing frequency of cranial nerve 8. When the stereocilia tilt away from the gynocelium, the hair cell is hyperpolarized, decreasing the amount of neurotransmitter released, which decreases the frequency firing of cranial nerve 8. Now, this is the vestibule to which is attached is the utricle and the saccule. Now, inside the utricle, we have several stereocelium, and supporting it are the supporting cells. The hair cells of the stereocelium are embedded in a gelatinous matrix, and embedded within the gelatinous matrix are the otoliths. And the otoliths with the gelatinous matrix combinedly form what is called the autolyth membrane. And these are the autoconia, and a single stereocelium at the terminal end of which is the vestibular nerve branches. And we have one long hair cell, and that is called the gynocelium. Again, in the utricle, we have a structure flattened 2 to 3 millimeter is the macula. And in the macula, we have the hair cells, the supporting cells, the otoliths, the gelatinous matrix. The autolytic membrane is composed of calcareous material embedded in a gelatinous matrix. This is the autolytic membrane, the gelatinous matrix with the autoconia. Now, this has a mean thickness of 50 micrometers. The position of the autolytic membrane on the sensory receptor depends on the magnitude and direction of force acting on it. Even when the head is at rest, the calcareous material, because of its mass, exerts a force on the receptor equal to the product of its mass and acceleration due to the gravitational pull of Earth, which at sea level is 9.8 meters per second squared. The distribution of force acting on the underlying sensory cells changes with different degrees of head tilt and linear head accelerations. The utricular macule is located next to the anterior opening of the horizontal semicircular canal and lies mostly in the horizontal plane. In contrast, the saccular macule lies in a vertical plane in between that of the two vertical semicircular canals. This is the autolytic membrane comprising of the gelatinous matrix and the autoconia. And deeper below we have the hair cells and the supporting cells. At the edge tip of it are the stereocelium and the gynocelium, which is one long hair cell. Let's have a look at what you mean by the supporting cells. The development, function and maintenance of inner ear sensory epithelium are heavily dependent upon supporting cells, which are non-sensory cells that reside between hair cells. Unlike hair cells which contact only the luminal surface of the epithelium, Supporting cells span the entire depth of the epithelium from the basal lamina to the lumen. Supporting cells are linked to each other and to hair cells by tight and adherent junctions, and they communicate freely with other supporting cells by gap junctions. Supporting cells serve a diverse set of functions in the sensory epithelium. They have rigid cytoskeletons that maintain the structural integrity of sensory organs during sound stimulation and head movements. Supporting cells also help to maintain an environment in the epithelium that enables hair cells to function. For example, hair cells recycle potassium ions, which help to maintain the driving force for generating the receptor potential. They generate components of the tectorial membrane in the organ of Conti, the autoconal membrane and autoconal components in the macula organs, and the cupula in the cristae ampullaris. Following trauma or toxicity, the supporting cells can eject injured hair cells from the epithelium, phagocytose hair cell debris, and in some cases generate new hair cells. Now, these roles of supporting cells will be discussed in detail at a later point of time. Supporting cells have another essential role in the repair of sensory epithelium after hair cell damage in non-mammals. These are emerging knowledge. They regenerate hair cells in both the auditory and vestibular epithelium of birds and reptiles, as well as vestibular and lateral line hair cells in amphibians and fish. In birds, regenerated hair cells restore hearing and balance function in a couple of months after damage. Humans cannot do that. Why? Because the gene for regeneration, ATOH, is locked. In vestibular epithelium and in lateral line neuromass of fish, hair cells undergo continual death and replacement in adulthood. And in the case where supporting cells continuously form hair cells throughout life, they are thought to have some stem cell-like characteristics. In mammals, the degree of hair cell regeneration that occurs after damage is limited. No hair cells are replaced in the adult organ of Conti. Why? But the gene for regeneration is locked. By contrast, a low level of hair cell replacement occurs in adult mammalian vestibular system following hair cell loss. Let's have a look. This is the vestibule in which we have the utricle and the saccule, and then in the utricle we have the macula, and in the macula we have the otolith membrane, wherein we have the gelatinous matrix and the autoconia. Now deeper below are the hair cells, in which there are multiple stereocilia and one long gynoecilia. When gravitational effect causes this to bend forward, like look at this person bending forward like this, the autoconia causes the gelatinous matrix to tilt forwards, and as it tilts forwards, these hair cells have a shearing effect and are pushed forward in order to generate impulses that are carried via the auditory nerve and then identify the balance changes of linear acceleration. The cavity of the utricle communicates behind with the semicircular ducts by 5 orifices. The ductus utriculosacularis comes off the anterior wall of the utricle and opens into the ductus endolymphaticus. The vestibule is located within the bony labyrinth of the inner ear. I repeat again, the utricle and saccula co-located within the vestibule. The utricle lies on the medial wall of the vestibule and is in contact with the recessus ellipticus. It is larger in size compared to the saccule. The utricle communicates with the saccule via the utriculosaccular duct. Lying horizontally on the floor of the utricle is a projection called the macula, the sensory organ of the utricle. This sensory epithelium is lined by vestibular cells, which are mechanoreceptors containing stereocelium and gynocelium. Now, these cells project into the membranous labyrinth called the statoconial membrane, a gelatinous layer beyond containing otoliths. Cilia from the hair cells are attached to these otoliths. Any change in the orientation of the head results in stimulation of the utricle via movement of the otolith cilia macula complex, releasing signals that the brain interprets. The utricle is more sensitive to horizontal acceleration in comparison to the saccule, which is more sensitive to vertical acceleration. The anterior vestibular artery is the second branch of the labyrinthine artery, supplying it. The mechanoreceptors of the utricle are connected to the nervous system by fibers of the vestibular division of the vestibulocochlear nerve. The utricle is too small to be distinguished on high-resolution CT or MRI, but sits in the medial portion of the vestibule. The utricle, again, we repeat, contains mechanoreceptor hair cells that distinguishes between the degree of tilting of the head thanks to their apical stereociliar setup, covered by otoliths, which due to gravity pull on the stereocilium and tilt them. Depending on whether the tilt is in the direction of gynocelium or not, the resulting hair cell polarization is excitatory or inhibitory. Any orientation of the head causes a combination of stimulation to the utricle and saccule of the two ears. The brain interprets head orientation by comparing these inputs to each other and to other input from the eyes and stretch receptors in the neck, thereby detecting whether only the head is tilted or the entire body is tipping. The inertia of the autolyth membrane is especially important in detecting linear acceleration. Suppose you are sitting in a car at a stoplight and then begins to move. The autolyth membrane of the macula utricle briefly lags behind the rest of the tissues, bending the stereocilia backwards and stimulates the cells. When you stop at the next light, the macula stops, but the autolyth membrane keeps going for a moment, bending the stereocilia forward. The hair cells convert this pattern of stimulation to nerve signals, and the brain is thus advised of changes in your linear velocity. The next time when your car suddenly stops and you jerk forward, imagine what is happening to the autolyth membrane, the gelatinous matrix, the autocornea, and the stereocilium and the gynocelium. Now, this signal to the vestibular nerve does not adapt with time. The effect of this is that, for example, an individual lying down to sleep will continue to detect that they are lying down hours later when they awaken. Unbent and at rest in the macula have a base rate of depolarization of 90 to 100 action potentials a second. The brain suppresses this and we ignore it and know that our body is stabilized. If the head moves or the body accelerates or decelerates, then bending occurs. Depending on the direction of the bending, the hair cell will either be excited or inhibited, resulting in either an increase or decrease in firing frequency of the hair cells. The macula is also sensitive to linear acceleration, as the inertia possessed by the statocornea can also shift the gelatinous layer during increase and decreases in linear velocity. Finally, the human utricular hair cell number is about 33,000 compared to the turtle, which is 8,000, and the mouse, which is about 3,000. However, the hair cells per unit area are somewhat similar, with humans of 7,000 hair cells per square millimeter, whereas in the turtle is about 10,000 and the mouse about 19,000. So here we have an overwhelming overview of the utricle, and I hope you found it useful.